Right. So this is my great pleasure to welcome today Alan Love and Günter Wagner. They have been collaborating for several years, and this is a very interesting combination of philosophy and biology, especially about developmental biology and Evo-devo. As you probably all know, Alan is John and Dolan Professor of Philosophy at the University of Minnesota and Director of the Minnesota Center for Philosophy of Science, which is really one of the most important, I think, centers in philosophy of science in the world. And the research of Alan focuses on conceptual issues in biology, and his work has focused on Evo-devo, developmental biology as such, molecular biology, and paleontology as well. Günter Wagner is professor of ecology and evolutionary biology at Yale University in the US as well, and at the Department of Evolutionary Biology at the University of Vienna. His main research interest is in the evolution of gene regulation as it pertains to the origin of evolutionary novelties. Both Alan and Günter are members of the PhilInBioMed Network that puts many of us together, as you know. What they will present today is based on a paper with James DiFrisco, and James now has his team at the Crick Institute, a team focusing on theoretical biology. This is called Theoretical Biology Laboratory. But I'm not going to say more about this because I know that Alan will mention that again in his presentation. So this is really our great pleasure to have the two of you, the three of you today, Alan, Günter, and James. And the title, I'm just trying to find the title, The Hierarchical Basis of Serial Homology and Evolution in Novelty. So the floor is yours, then thank you again for accepting our invitation.
All right, thank you, Thomas. I'm sharing the screen now. I'm assuming it's coming through okay.
It's
please
perfect.
let me know otherwise. Okay. Yes, and thank you again for the invitation, Thomas, for being here. And we're excited to share this work and hear perspectives on it from those who have joined us. So this is joint work with James as well. We're giving James a break today on the presentation, but for those who are interested, this is the paper that came out earlier this year in the Journal of Morphology. What we will be talking about is only a portion of it. There's lots of things that we won't be able to include because of time. It also, in reference to Thomas' point about the collaboration, emerges out of work over the last several years that James, Günter, and I have done on character identity mechanisms, exploring different angles. We will not go into the details of all of these different things today, but we just wanted to let people know this and also to highlight because in many ways it exemplifies philosophy in biology and medicine in the sense that the collaboration is an intentional mixing up and combining of philosophical and biological themes with the aim of advancing theory and understanding in biological science more broadly, but also we think nicely illustrates certain forms of interdisciplinarity that can emerge in communities where philosophers and scientists regularly are interacting. And in particular, I do want to highlight that James is himself a great example of this, somebody who was trained in philosophy and through his postdoctoral years working in different philosophy units, but actually landing a permanent position at the Crick Institute as a group leader in the theoretical biology laboratory. This is quite an achievement, and I think is a nice testimony to the kinds of work that can be done across standard boundary lines of disciplines that exist out there today. Okay, so here's what we want to do in the time that we have. We want to set up a broad problem for homology. We want to talk a little bit about the strategy that we adopted in trying to tackle that problem, which we call theoretical articulation. And then we will introduce the ongoing question of serial homology. Then we'll talk a little bit about character identity mechanisms to give some of the basics that are needed for our presentation, which will primarily culminate in the discussion of hierarchical serial homology, where we think we can offer some new distinctions and vocabulary as a consequence of those theoretical resources. We'll briefly point out some applications to evolutionary novelty before then concluding. So let me begin with a problem for homology. We've now got two well-established empirical generalities. One is the pervasiveness of gene sharing in evolution, something that has been documented repeatedly and in a number of different systems across the tree of life. And we also know that there is homology across the tree of life. That is, ways in which characters are shared, and sometimes shared quite deeply in phylogenetic relationships. But together, these form a problem. Given the fact of common descent, why is everything not homologous with everything else? If there is this pervasiveness of gene sharing, this pervasiveness of sort of interconnection across the tree of life, how do we decide when something is homologous versus when it's not? Because if we can't make that decision, then the category really doesn't have the bite or the traction that it needs to for ongoing empirical investigation. The composition and developmental formation of traits at different levels of organization across taxa are affected by this extensive gene sharing, and that makes it difficult to discriminate, especially at the genetic level, one trait from another. And drawing a sharp contrast between traits that are homologous and others that are not seems to rely on selectively ignoring some portion of this ubiquitous common inheritance. And if we're selectively ignoring it, are we doing it just because of a personal preference, or is there some good reason to do so, some theoretically grounded account of that? So the continuity and overlapping genetic and developmental contributions to diverse traits across lineages seems to imply that no discrete determination of homology is possible. Now, this is a problem with some interesting details that also have emerged over the past few decades and are especially acute for hierarchical notions of homology, which zoom in to focus on the developmental mechanistic basis of characters at different levels of organization. Developmental mechanisms for traits such as eyes and limbs are more conserved than initially thought, exhibiting substantive similarity across wide phylogenetic distances, something commonly referred to as deep homology. At the same time, homologous traits are induced by different signaling pathways in different species or arise from different germ layers and cell populations, what's often termed developmental system drift. So you have both more conservation than expected and more diversity than expected. Some have argued that this widespread overlap in parts and processes should be acknowledged as partial homology, since most characters share some genetic and developmental commonality. That's going to lead to a dilemma, though, because if that's true, then partial homology is always going to be something you can detect. And then what becomes homology and non-homology seems to blur, and that threatens a broad base of comparative morphological knowledge assumed to be robust by most biologists. So really what we want to do in this paper, and the motivation, was to find a way to avoid that conclusion that somehow everything is homologous with everything else. On what basis would we be able to come to this conclusion, given we now have these empirical generalities that we are facing? And so that led us to a particular strategy, a strategy we call theoretical articulation. Biologists are often faced with difficulties in conceptualizing phenomena because new data arrives rapidly and thereby contributes to the disruption of theoretical categories. In this situation, biologists adopt a strategy that involves introducing distinctions to extant concepts, sometimes revising or narrowing their original meaning to produce increased contrastive resolution among the labels used to represent biological phenomena. Examples from homology in particular can be seen by looking at the far past and Richard Owen, or more recent discussions of concepts like positional homology, organizational homology, or deep homology. Theoretical articulation is a strategy that involves articulating an elaborated set of conceptual relationships to yield productive theory for doing empirical work. It's not something that just falls out of our existing ideas. It's something that has to be developed. And importantly, the strategy can fail. Terms and distinctions do not catch on, or there are underlying difficulties with distinctions. And we can see this, in fact, in looking back at the history of homology. Distinctions have been introduced, terminology has been put forward and has fallen by the wayside and not been deemed to be helpful. Yet, when it is successful, the articulation can enrich investigation and come to undergird the basic vocabulary that's utilized daily by biologists. So that's what we are doing here, is adopting this specific tactic to try and understand serial homology. And what we're going to do is zoom in on successful patterns of reasoning about homology at the level of gene sequences and ask to what degree can this help us get traction on elaborating this richer set of conceptual relationships that will inform a better perspective on serial homology. All right, Günter?
Okay, I'm ready. Okay, so let's look at serial homology more specifically. And, as Alan already said, serial homology was actually part of the theoretical articulation that Owen has proposed. But in particular, in the second half of the 20th century, serial homology got into trouble, primarily because it was understood that real homology is based on phylogenetic relationships, but serial homology is about different parts in the same body, and therefore it doesn't seem to have a phylogenetic basis. So let's look at this problem, to this apparent conflict, a little bit in more detail. So homology in general is a hypothesis about the traceability of biological characters that is made possible by the fact that some parts of the body are developmentally individuated and therefore can be seen as units of evolutionary change over time. Now, emphasizing that homology is actually something about traceability in phylogenetic history seems to stand in contrast to the observation that homologies are often related to similarities. Now, it is true that homologies, in particular in morphology, are first discovered through similarity, but they are ultimately worked out to make sure that hypothetical, putative homologies can be put into a phylogenetic framework so that eventually we can be sure that these homologies are actually tracing a real evolutionary transition, evolutionary history that can sort of be seen as a way of tracing identities through historical time. So given this general characterization of homology, the question, as I mentioned at the beginning, the question is, does it or can it actually apply to serial homology, and namely the relationship among similar or corresponding parts of the same body, such as ribs in this mammal here. I think it's a human. So the one of the problems is that, you know, there is an intersection here between phylogenetic relationships and relationships among different parts in the same body, and that leads to the apparent contradiction that, you know, serial homology is both a relationship of parts within the body, but also serial homologs are shared across taxonomic groups, which, you know, raises the question, you know, how to, you know, integrate these two different perspectives. Next. So there are sort of three possibilities to deal with this fact. The one is to emphasize the phylogenetic perspective, and the way that works is you take serial homologs or repeated parts that belong to one particular group of parts, and lump them together into a singular trait, like the concept of the cervix or cervical vertebrae, and that group then is the thing that can be compared across species, and can be traced through phylogeny. So that essentially would turn serial homology completely into a case of special homology, you know, the one that we usually talk about. The other possibility is to take an intraorganismal perspective and observe that, you know, within the same body, often structures occur that are very similar and also developmentally equivalent, so that we cannot escape the conclusion that they are in some way the same thing repeated in the same body. So that would then completely ignore the phylogenetic dimension, and therefore is problematic in itself. So what we want to do is to actually provide a perspective or a theoretical articulation, as Alan said, that would be able to, you know, combine both of these perspectives. Okay, so we did this by proposing, you know, what we can say a new concept or a new way of articulating serial homology, and that says something like this: two characters in the same organism are serial homologs if they are derived from differentiation of a single character in an ancestral species, or a set of identical characters in the ancestral species. So the implication of this is that even serial homologs that are in the same organism are still related to each other through an evolutionary process of differentiation. So that's the way how we sort of want to you know, square the circle that is intrinsic in this notion. And of course, we haven't invented this way of thinking out of nothing. Of course, you know, I was inspired by the work in molecular evolution, genomics in particular, where it's, you know, in the mid 20th century, it became clear that there are genes in the same genome of a particular species that are related to each other through gene duplication. So an early example are the different peptides for the globin gene, so the alpha chain and the beta are originated in evolution through duplication in the genome, and are now co-transferred from generation to generation, so that each of these two duplicated genes have their own evolutionary history that reflects the history of the species in which these two genes are present in the genomes. And if we have genes that are arose through duplication and are in the same genome, we call them paralogs. And of course, paralogs is, you know, the molecular equivalent to serial homology, because, you know, it's just a repeated part of the genome and therefore part of the organism, in the same way as morphological characters potentially. We call the same genes in different species that are derived from the same duplicate as orthologs, that ones that are corresponding to each other originating from genes as paralogs, as I read, and all of them are different forms of homology, essentially. So that's one other conceptual tool that we can lift from the molecular evolution side of things is that if there are more than two paralogs, then we have sort of a larger family of genes that are related to each other in a hierarchical way, the same way as species are. And then it is useful to call them a gene family. And we will do the same thing for morphological characters. So as I said, paralog genes are a kind of simple example of serial homologs, or the most basal molecular instantiation of this problem of repeated polyparts. Next. So of course the critical question is whether we can scale up from DNA sequences and genes to organismal units that are more interesting nowadays. So our example are cell types. And cell types, as you can see, are blood cells, epidermal cells, bone cells, and so on, and muscle cells, neurons. Of course, across species can stay in the relationship of homology. For instance, the skeletal muscle of a cow is certainly homologous to the skeletal muscle in a human. I'm now referring to cows because I'm here in Texas, working with people who are working on farm animals. But there's also an observation that there are in, let's say, a mammal, different muscle cells, and they also relate to each other in the following way. For instance, it turns out that heart muscles cells, cardiac muscle cells and smooth muscle cells are actually more closely related to each other than either of them are to skeletal muscles, even in apparent contradiction to the similarity, phenotypic similarity, where cardiac muscles and skeletal muscles are called striated, but in fact, evolutionarily and developmentally, they are more closely related to smooth muscles. So this is encourages us to look at cell types in a similar way as we are used to do that with genes. So these cell types, then you can argue are cell type families. So all muscle cells in a mammal are related to each other, but within this group have a certain phylogenetic structure. And of course, neurons, many of them, not all of them, but most neurons are related to each other, but pyramidal neurons in the cortex are more related to each other than they are to granular ones in the Purkinje, in the cerebellum, and so on. So this is a evolutionary perspective on cell type identity that Detlef Arendt and friends and I have articulated in this paper in Nature Reviews Genetics in 2016. Okay, so how to think about this a little bit more in detail. So here is, sort of a cartoon species phylogeny where we have a yellow, a blue, and a red species. And this, tree here is the phylogenetic relationship among these the species. And within this species tree, we see different lines that represent the inheritance of particular cell types. So we have in a stem lineage here, we have one cell type that then differentiates into different cell types that are passed on to their descendants, and then they can undergo further diversification, just in the same way as genes that are duplicated can further duplicate and create whole families of genes. So we have the same here. So if we pull this cell type tree out of the species tree, we get this, what we call the cell type tree, which reflects both evolutionary origin of new cell types, like this here, or this here, as well as the phylogenetic relationships among the species which actually have these cells in their body. Next slide. Okay, so this leads to this concept of a cell type tree. Here's an example, a paradigmatic example. Here in the case of ciliary photoreceptors, there's a parallel tree on the other side of rhabdomeric photoreceptors. And what that suggests is that, for instance, the rod cells are more closely related to one subtype of cone photoreceptors rather than other cone receptors there. And what is even more important here is to realize that not all of the terminal branches here in the cell type tree, even though it's a cell type tree of photoreceptors, need to be photoreceptors in a physiological sense. A good example is this bipolar cell, which is an interneuron in the retina, but evidence very clearly shows that it is a modified photoreceptor that originally was a genuine photoreceptor, but then specialized on the function as a interneuron. And from a phylogenetic point of view, this cell, even though physiologically not being a photoreceptor, although there's some residual ability of it to react to light, is phylogenetically seen, still belongs to the family of ciliary photoreceptors. So homology doesn't mean that the function remains the same. And then you have this. Anyway, let's go on. So this idea that cell types are related by a phylogenetic tree is underwritten by this idea of sister cell type origination, meaning that new cell types arise through differentiation of a single cell type in the ancestor. And it's of course hard to know how to actually test this idea in a direct way. But what we can do very often, the only way what we can do in much of empirical science is to just test predictions of the model rather than the model itself. Next slide, please. And the opportunity to do that comes from this idea that new cell types arise from a common ancestral cell type by gradual differentiation, where at some point the gene regulatory machinery starts to diverge, here indicated by the red and the green transcription factor 1 and 2, that gets either expressed in the one or the other cell population. And then that leads to either the loss or recruitment of particular functional modules in the cell that eventually leads to a gradual differentiation of cells in the transcriptomic profile. Now, if that is the case, we should actually see a trace of that in the expression of genes among cell types, and this is what we tested in the next slide. So if that is the case, cell type transcriptome should have a hierarchical structure, and that hierarchical structure possibly reflecting the evolutionary history of the cell types. Now, in order to test this, we took advantage of the fact that our colleagues in molecular phylogeny or comparative biology developed statistical methods to test whether any set of taxonomic units, as they say it, in their sequence similarity or other characteristics actually have a hierarchical structure. Basically, the question whether any data has a phylogenetic signature in it. And one of these tests is the so-called quartet test, treeness test, so you can test whether there is some. And the idea is you can always take 4 of these species or OTUs or cell types or whatever, and calculate the similarity, and their relationships can always be represented in such a box diagram. And it is obvious if this inner box, the E and the F, are very unequal and E eventually collapses, we have a tree-like structure if E is much smaller than F. And that is sort of the basic idea. We developed a null model for gene expression data, presence, absence. And with that, we can sort of calculate how likely it is that a particular pattern of similarities arises by chance, or whether it is some kind of deterministic process that produces treeness more likely than by chance. Next slide. So we tested this with a number of cell transcriptomes that were available to us at the time. This is work done by Zhong Liang, at the time a postdoc in my lab. Actually, I was a graduate student, sorry. And this is the distribution for all quartets of cell types that we had of these sort of p-values or alpha values. And if this frequency distribution is highly biased to low values, that is a very strong statistical indication that this data set has a very strong signal for a hierarchical structure. Here you have another way of representing this, and here in a cladogram. So from a statistical point of view, it is clear that cell types in the body, this is mouse and human cells, have a very strong hierarchical structure, which is consistent with, but of course no direct proof of the sister cell type model. Interestingly, we also did this for cancer cells, and if we do cancer cell similarities, it turns out there is no hierarchical structure there at all, which shows that cancer cells are the product of a breakdown of gene regulatory well-behavedness. Okay, next slide. Okay, so I think cell types are relatively good bet of to behave in a similar way as gene duplicates, but how or whether we can scale it up to multicellular parts of a body, namely, is the next question that we want to address. One nice example that we talked about are epidermal appendages, hairs, feather, scales, and so on. And we would argue, and actually other people also have argued, that they are also phylogenetically related to each other in the same way as genes and paralog genes and different cell types in a body are. So next point here. So for instance, a mammalian hair is not just a keratinous shaft that sticks out from the surface of the skin, but is actually a relatively complex microanatomical structure with glands, sebaceous glands, pulps, muscles, and so on. So it's actually a quite complex structure itself, and that is also the basis for the fact that hair can also give rise to much more impressive, functionally impressive structures like mammary glands in mammals. It turns out that mammary glands are in fact phylogenetically derived from a group of hair, and the sebaceous glands and or the sweat glands probably conspired to turn into mammary glands. And this hypothesis that mammary glands and hair are essentially serially homologous structures is also supported by the fact that, for instance, opossums, in early stages of the differentiation of the mammary gland, it still has a hair sticking out, which then falls out. Interestingly, that's also the case in horses and in a few other eutherian species. So in some of the species, mammary glands during the development still betray their relationship to hair by temporarily producing a hair that then falls off later in development. Next slide. So if we follow this logic, we could, here have a very rudimentary tree of skin appendages. There's certainly more to be said about it. But in the mammalian case, so we would have a hypothetical origin of the this sebaceous unit, and then there would be different forms of hair, like contour hair and underhair, or whiskers and so on at the one hand, but all of them would be phylogenetically related to mammary glands, which pretty certainly are an elaboration of this PSU. On the other side, the reptilian side and birds, we have scales and feathers, and it's also pretty clear that feathers derived in a certain way that I cannot talk about here from changing the development of scales. So they seem to be more related to scales. By the way, real scales we don't have in mammals, so that also supports feather scale relationship. Next slide. Okay, so so having made an argument that there are that serial homology can be thought of as evolutionary through the lens of evolutionary differentiation, we have to point out a few things that we need to keep in mind. First one, so for instance, repeated cellular elements like neutrophils, I think those are neutrophils, these are certainly erythrocytes, and we wouldn't call them serially homologous because they are simply copies of the same developmental program, reiterated again and again during the lifetime of an animal. So this is simple copies that is not the same as, and we will elaborate this a little bit more, as serial homologs. And there is also an example at the molecular level, in particular the ribosomal RNA genes, and in the 70s, 80s, it was discovered that the different, obviously parallel copies of the ribosomal genes in one genome are much more related to each other than any one of them to the ribosomal genes in another species, which means that you cannot trace the history of a particular ribosomal gene through a common ancestor to another species. And that's because these ribosomal genes are under a process of constant turnover, so that new copies are produced at a high rate, and they are lost also at a high rate, so that the set of ribosomal genes is essentially a population of, if you want, population of genes that exist as a group, but its individual members have a very shallow evolutionary history. So those is an example, are examples of sort of mere copies at the molecular level, not only at the cell level. But on the other hand, repeated elements that morphologically are highly similar can be nevertheless evolutionarily individuated. So high similarity doesn't mean it is simply a copy. And one example would be fore and hind wing of a honeybee. They are looking very similar. Basically a blade of cuticle with veins in there, but they are clearly developmentally individuated, which you can already see here because they have a different size, and also the evolutionary history across pterygoid insects also indicates that fore and hind wing are actually individuated characters. Next slide. So that these complications show that it's dealing with homology and serial homology is quite complex, and there is a lot of confusion in the literature about their relationship, and therefore we thought that in order to get through that and make it more productive, we need more of theoretical articulation in order to not lose that part of comparative biology at all completely, because people don't know what to think about it. With that, I'm handing over to Ellen again.
Recall our problem. We've got similarities and differences that can be found between homologous characters that we expect from the long-term evolution of independent lineages. And we're trying to make sense of how a notion of homology and a definition of shared characters abstracts from that biological variation and diversity, picking out what is invariant or conserved in a theoretically appropriate way, but also recognizing that we have to figure out what to pay attention to and what not to. Sameness is not similarity. And if we didn't do that sort of sifting of different features to compare and pay attention to, we wouldn't be able to make comparative generalizations. We'd be in that position where everything would be homologous with everything else. So that brings us to an important sort of baseline for the theoretical articulation, which is homology as sameness. To say that homology is between two characters means they're the same kind of character, and that this kind forms a historically traceable individual. And thus, homologs can be highly dissimilar. Dissimilarity on its own is not somehow an indicator that things are not homologous. However, judgments of homology are binary or discrete. Two characters are either homologous or not, as opposed to having some degree of homology. And that yields equivalent classes of characters that then can enter into comparative generalizations. It's not partial homology, and that might be a temptation in the face of the biological variation and complexity. But in fact, it would just shift the problem, because degrees still require specification of the properties that living systems can have degrees of. So we just need to remember that the existence of different degrees of evidence for homology does not entail different degrees of homology itself. And so that brings us then to character identity mechanisms as a way of undergirding a notion of sameness mechanistically. Character identity is not just a morphological fact, but also something controlled and explained by distinctive causal mechanisms that operate in development, with consequences for evolutionary persistence and traceability, giving you both the developmental and the evolutionary individuality. And character identity is contrasted with mere similarity between characters and with character states. You can have thematic variations that characters with a given identity exhibit, such as size and shape, but they can still have the same identity, namely, say, something like a wing. And the causal profile that exhibits during development can be represented in the cartoon form this way, where lots of different kinds of initiating signals can contribute to the operation of a character identity mechanism, which then can also lead to divergent outcomes consistent with that same identity. But the causal profile of the chim itself is quite refractory to too much change. And in fact, when experimental interventions occur on character identity mechanisms, we often get catastrophic failure where any version of the character simply doesn't emerge. Now, importantly, character identity mechanisms exhibit different features at different levels of organization. So we don't expect that the mechanisms will be the same. And we won't have time to go into the details on this, but this just gives you a snapshot of thinking about how we can understand character identity mechanisms hierarchically, in the sense that they are not going to exhibit the same features necessarily on each level of organization. And so this is just a picture of what you might expect from cell-cell signaling interactions that support a tissue type. And the similar sorts of things can be done at the cell type level, which you already heard Günter talk a little bit about in terms of regulatory networks. And as well, you can move to consider this at the organ level as well. Now, the important thing for our purposes is recognizing the character state possibilities in looking at serial homology. You can have differences in the configuration of subparts. You can have differences in attributes, such as color, and the parts of a character are themselves entities. potentially characters, but having such and such parts is an attribute of the character. So here we have to be very careful about our theoretical distinctions. Characters can comprise attributes such as feathers, themselves biological characters, but the presence or absence of feathers in tracks is an attribute of the organism's phenotype. And those differences in character state can include both differences in attribute or differences in kind and number of subparts. And those are the things that we will need to pay close attention to when we look at how to distinguish different forms of iterated parts and whether or not they count as serial homologs. So though that is quick, we now have basically what we need to articulate a more precise account of serial homology. And I'm going to turn things back over to Günter to walk us through the basic scheme.
Okay, thank you. So what is coming now is a little bit could look like an overbearing scheme of difference and differentiations that seem to be more semantic, as some people say it. But having been in the discussions about homology and serial homology for the better part of a half century, I have to say that a lot of the inability to communicate in these areas arises from the fact that we are using a language that at an intuitive level covers quite different biological situations. And if our language doesn't reflect this biological differences, we are making no progress. So we are trying, James, Ellen, and I have tried to come up with a scheme to articulate clearly what kind of biological situations we are talking about. Now, at the lowest level, we often talk about character states attributes and compare those among species, for instance. And by attribute, we mean things that are descriptors of the character, but cannot be in a way separated from that, like shape or color. Okay, you can, let's say structural color, you cannot actually separate. Pigments you couldn't separate. But anyway, so there are the similarities of character attributes, and it turns out that I think these character attributes are very hard or only traceable in a very limited way across species individually. Okay, next slide. Then there are cases where you have names for parts that are sort of anatomically separable, but they are not, as far as we can tell, own their existence to a independent character identity developmentally, so they have no gene supposedly, that are more individuated by descriptive context. One example is this zygomatic arch here from this post-orbital bone to the petrosome. And this arch here you find in all the mammals, but its relative contribution from the one bone or the other can be highly variable. So this is actually sort of a spatial relationship of various parts there. Like the same as the transverse spinous process, probably doesn't have its own developmental identity. You can talk about it, how it develops, but that is, or we can have various veins in the periphery of the circulatory system, some of which you can actually trace from one individual to the next, but it's pretty clear that they are not arising through a body part specific developmental program, but rather from an interaction between a reaction norm of how blood vessels develop depending on blood flow characteristics and the anatomy in which these or the spatial relationships they are finding themselves. This is what we call sort of character state structure. And it is related to what Gerd Müller called organizational homology, where the homology and individuality arises from the context in which a structure can be found and thereby traced in a contextual way. Next slide. Then we have this case that I mentioned before, with the body parts that are simple copies of itself, like different contour hairs in the fur of an animal, different instances of the same cell type in the blood or in the body in general, or leaves, for instance, in a plant. In this case, it's further the case that we cannot even trace individual instances of these structures across individuals, or not to speak species, because they are essentially the same thing, and they have a very variable or no spatial context in which you can find them reliably, and therefore they are absolutely not traceable, not even among individuals from the same species, not even between father and brother and son or so on. Then there's a situation where we have parts that clearly are the result of a particular developmental program, let's say a digit, but they are also just copies of each other. So there's a lot of evidence that this is digits, index finger, middle finger, ring finger, and pinky, are in humans and in many other mammals not actually differentiated. So they are in a way similar to the replica, the copies that I talked about before, but there is a certain degree of traceability because their spatial localization in the body can be identified. So this is what we call positional indexing. So it is tempting to talk about 2, 3, 4, and 5 across different animals, and they are traceable because of their positional indexing, because their positional context is quite conserved. But this traceability breaks down if the spatial context gets disturbed. For instance, if you lose, let's say, one digit, then you have great difficulty actually finding out what the remaining digits are, because the spatial context is lacking and they are not developmentally individuated either. So there is traceability, but only as long as the positional context remains constant. Next slide. So this we call, actually, as a previous one we called homomorphs, and now we come to our paramorphs. Those are body parts with distinct developmental programs, endowing them with developmental identity, and have positional indexing, like the 3 thoracic segments of insects, thorax segment 1, 2, and 3. And they have their own genetic machinery that directs their development. Different parts of the differentiated parts of the vertebral column, like thoracic, lumbar, and cervical vertebrae, and so on. And digit 1 and digit 2. Digit 1 is actually quite, to the thumb, it's actually quite distinct and also traceable in its developmental distinctness across amniotes, at least, as far as we can. So we call those paramorphs. So among those sort of characters, we realize that there are areas where the similarity is either morphologically or mechanistically underwritten. Next, please. So this is our whole table here, where we try to make sure that if we talk about similarity of different parts or their relationship in the same body, we remind ourselves to always try to identify whether what we are calling similar or corresponding parts in the same body, what kind of biological situation that is reflecting, whether it's just an attribute that is and something that is attached to the character itself, so being the same color or same curvature, same number, separate structures, replicas without positional indexing, replicas with indexing, which we call homomorphs, or paramorphs that are, the individuated characters that we usually would call still homologs. Okay, and that have a phylogenetic structure. Okay, so how does this help us get some clarity on the notion of evolutionary novelty? So the question that is often asked is, are serial homologs more of the same or something new? Remember that we limit our notion of serial homologs to exactly those that are individuated. For instance, the first digit, the thumb, is a novelty or distinct characteristics as when it arose in sometime in the tetrapod clade from the others. So they are because they are developmentally differentiated in a way that is constant and traceable compared to the more post-axial digits 2 to 5. So they are both, they are serial homologs in the sense that they are the same as digits, but in contrast, but the digit 1 is differentiated from the rest. But if we have a supernumerary digit 6 digit, which can happen pathologically, it would be a homomorph because it's probably just a replication of the digits 2 to 5, and that would not be a novelty in itself. Okay, go on. So from this perspective, differences of a common, so our perspective sort of tries to overcome a misguided assumption, namely the question that that novelties have to be completely, coming out of the blue without any pre-existing structures or mechanisms to be related to. Like Shubin has articulated, 2 new anatomical structures arise de novo, meaning there's no pre-existing structures at all. But I think this perspective is not tenable because of the pervasiveness of gene co-option, reuse of existing structures, and so on. So we have to focus on the question of whether a structure that is hypothetically a novelty, whether it is a novelty through the evolution of novel individuality of the body part compared to what was present in the common ancestors. Yes. So we are not, so our theoretical articulation then takes into account that characters are hierarchically related to each other, even within the body. So the reuse of pre-existing structures, mechanisms, and pathways is not a problem for us as identifying something as a novelty. But the relevant question is how that is happening. So then the question when we judge a particular derived structure, traits, characteristic as a novelty or not, does not depend on the degree to which pre-existing mechanisms, structures, or cell types are used in this putative new character, but the question is whether the configuration that we identify is due to the developmental individualization at a particular hierarchical level of organization. And therefore, it's important to make this distinction between replicas, homologs, paramorphs, and at a particular hierarchical level. Next.
Okay, so let me then just wrap us up really quickly with a few conclusions, and then we can go to questions. So remember that we had argued that the goal was to offer a theoretical articulation that helped advance investigation. And we think actually this is something that's been recognized in the past by other researchers. So Ray Lancaster sort of recognized this with respect to Richard Owen, that it's not easy to exaggerate the service rendered by Owen to the study of zoology by the introduction of this apparently small piece of verbal mechanism. And though the conceptions of archetypal morphology to which it had reference are now abandoned in favor of a genetic morphology, yet we should remember in estimating the value of this and of other speculations which have given place to new views in the history of science. So we sort of see ourselves in this vein, trying to offer verbal pieces of verbal mechanism that will advance inquiry. And we think one of the criterion for that is fruitfulness, the capacity to engender new forms of scientific inference or research, especially in the context of new empirical discoveries that disrupt current biological understanding, and that motivated us throughout. We use successful patterns of reasoning that have coalesced in talking about homology at the level of gene sequences, with some important theoretical desiderata to offer a hierarchical phylogenetic characterization of serial homology in terms of a five-fold theoretical classification. And I won't go over it again, but just as a reminder, this is that five-fold structure that we're offering that is beyond where we currently stand in thinking about how to look at and investigate serial homology. Our proposal is grounded in earlier work that elucidates what it means for a part to have identity as a biological character, and it's amply illustrated by findings over the past decade, especially in the studies of the origin and evolution of cell types. The theoretical elaboration, therefore, meets the criterion of fruitfulness and offers biologists a richer and more precise vocabulary, opening new avenues of inquiry. And with that, we'll stop, say thank you, and we look forward to your questions.
Thank you so much. That was a fascinating talk, and I think a fantastic illustration of really putting together philosophers and biologists and suggest new ideas in the way you just presented. Okay, let me switch maybe to the gallery view so that we can see everyone on screen. So we have about 15 to 20 minutes for questions. So I think it would be great if people could introduce themselves before asking a question. And by this, I just mean you say if you're a biologist, a philosopher, or a medical doctor, we don't need a full introduction or full CV, just it's important to see where the question comes from. So please raise your hand if you want to ask a question. I can start because I see that people are still sort of digesting this incredibly rich talk. So again, thank you so much. One question I have, which is pretty general, but I think very important, is to what extent in the end, the suggestion is to reform the language as opposed to making a novel theoretical suggestion, because this is really something you mentioned several times. So we're talking about something where we clarify the language about homologies, which I think is super important. Or are you doing something which is, for example, saying, if we're right, then these are the predictions we can make on the basis of our theory or our theoretical take on this question. And you mentioned that question, for example, when you talk about cell types and the extension to something else, but I just wanted to hear more about that. Are we more on the vocabulary side or the theoretical side, or it's the same, or it's very much connected?
Günter, you want to go ahead first?
Yeah, so my first reaction is that, what we try to do is, as a theoretical articulation, is, in my view, not the same as a theory as we understand it in the natural sciences, that, has a certain, a set of an ontology and principles from which you can make predictions, but it's actually, in a way, both at the language side, but also more importantly, it's the aim is to make our questions more precise, so that the precision of that question can be turned into an empirical question eventually, right? So I would not say, I mean, my example of predicting this, hierarchical structure of cell type transcriptomes does not derive from the notion of homology per se, but is derived from a particular model of how we think evolutionary changes in cell type identity occurs. This is the sister cell type model that was invented by Detlef Arendt. And from that model of evolutionary change, we can make predictions and then test them empirically. But that is sort of downstream of what we have tried to do here. And I would ask Alan and James to correct whatever I've said wrong here.
No, I would only add that the choice of the word elaboration is very intentional. So the idea is that we're building on existing theoretical resources. Some of those have been around for quite some time. Some of them are part of the work that we've been doing over the last few years and thinking about character identity mechanisms, and that we're building off of those to elaborate this new vocabulary, but also fully recognizing the historical point, which is that the proof is in the working out of these ideas in research. And so in that respect, none of us are going to sort of die on the hill of retaining a particular term, because the approach here is not to somehow preserve a calcified semantic framework, but rather to offer something that researchers can make more precise and refined claims with and test them better. So,
James, anything you would like to add for that specific question? Or maybe I should have asked before, maybe there's something you wanted to ask about the talk in general.
I just would add quickly that by connecting the notion of serial homology to this phylogenetic perspective on character identity mechanisms, that comes together with a bundle of specific hypotheses and predictions that when you observe morphological variation of a certain sort, or a character exhibiting kind of capacity to take on variations independently of other body parts, that it should have this underlying developmental architecture. And that's not true by virtue of the way the terms are defined, and could possibly be false, right? So that is a specific qualitative type of prediction.
Yeah, thank you for that. Any questions, comments? I'm sure people have something to say about this great talk. Okay, Richard.
Thank you. Let me put my hand down whilst I remember. Thank you guys so much. There's so much to think about there. My understanding is that the basic problem is, when is more different? Like if I have one character that has a particular way of being parameterized, and in one particular instance it has a value of 1, in another particular instance it has a value of 1.1, and we say, okay, well, that's just the same character, but it was just sort of continuously parameterized a little bit. But then, how far can we take that before it's something different? Is 2 really the same as 1? Now that I have 2 limbs instead of 1 limb, that's not really the same thing. That's something different. And I don't know if this is the same point, or whether I'm just rambling, but I'm thinking about character identity mechanisms, and whether you can really tell whether something is created by the same character identity mechanism or not. So, how when you fly around in the Mandelbrot set, you find areas which are black and areas which are white, and areas look like Seahorse Valley and areas that look like cartidoids, and all sorts of different shapes in there. But all of that complexity is created from exactly the same program. And it's a very simple program. And all that's really happening is that you're changing the parameters of the input, and you get completely different kinds of things on the output. So to say that something was produced by the same character identity mechanism seems like, well, at the very least, it's going to be very difficult to ascertain whether it was or not just by looking at it, right? And the details of what you find when you examine its internals are going to be different in one case and another case just by giving it a different input. Does that trouble you?
No. I mean, on a general level, I think as biologists, we have to get used to the fact that we are dealing with complex, historically generated systems, entities, right? And, I think the same problem arises, when are two populations from a different species or not? That's also, a gradual process. But what's characteristic of this example is, and I think it's also can be lifted over to homologs, is that yes, evolution proceeds mostly through gradual changes, but some gradual changes lead to absorption boundaries, in order to use a language that may be closer to your heart. And once you are there, there's very little chance to go back and mix them up again. And then these two things become independent evolutionary units of change, be it a species or different body parts. And of course, it's the question of how do we empirically know whether two parts of the body are generated or related to subscribed by different character identity mechanisms is one that leads us away from the conceptual issues that we discussed here. And I can give you examples if you want to get bored of how to go about it. One criterion is that genes that let's say contribute to a particular cell type identity seem to be necessary for that cell type identity, not necessarily expressed in others having the same function there. They also need to be evolutionarily traceable, as I said, so they need to maintain their individuality in a long phylogenetic time and so on. So there's a lot of downstream work that needs to be done in order to fill that out in any particular way. And maybe the last thing I want to say is that I don't think that biological diversity is a part of the Mandelbrot set, but actually a different kind of animal. But maybe I should stop here.
So I will add something complementary to what Günter said, which is that one might be concerned, Richard, in thinking about what sorts of mechanisms can produce identities. And if you can have, say, generic kind of mechanisms that yield diverse morphologies, for example, should we think about those as having independent identities? And I sort of bring this up in part to highlight that this is part of an ongoing set of papers that we're still working on. We don't think of ourselves as somehow having finished. And one of the things that we're currently thinking about actively is how to understand generic morphogenetic mechanisms, so-called Turing reaction dynamics and things like that in the context of character identity mechanisms, because we were not trying to claim that these things were not somehow relevant, but we were developing an account based on what we knew experimentally, at least from some intervention work that especially the cell type research. And then the idea is this elaboration is an ongoing endeavor, and we are continuing to think about these things, including the potential contribution of generic morphogenetic mechanisms and how they fit into the scheme.
Are there other questions, comments? get.
Unmute. Well, I think that the approach you're developing is a big step forward, but at the same time, it also contains some pitfalls, one might say. For example, if we argue that gene duplication and paralogy could be underlying serial homology, then in a sense, you're also arguing that molecular evolution drives phenotypic evolution forward. And I'm not convinced of that.
I agree with you.
And well, maybe I've misunderstood that, but
So yeah, so Gerd,
let me just say another
Okay.
pitfall is that, or could be, that if we are approaching serial homology via cell type evolution, this seems to slightly sidestep the original question of serial homology at the character level. And for example, vertebral serial homology, in which the cell types are largely the same in these serial homologs. So our pre-assumptions on how to approach the problem, in a sense, of course, as always, channel already the potential approaches that we might take. And I agree very much with Alan's final comment on how we need to link this in, or bring it together with the notion of generic phenotypic themes that can arise out of developmental properties in developmental systems.
So let me do 2 quick reactions and then let Günter follow that up. I think one thing that's really important is that the appeal to trying to get clearer on serial homology by looking at paralogy and gene sequence is not the same thing as a commitment that the higher level patterns are dependent on the lower level patterns. It's not a causal claim, but rather a conceptual strategy. The conceptual strategy to take what is being used in terms of distinctions at one level of organization and ask, can we work out similar distinctions at other levels of organization? The causal question will then be separate about the relationship between the levels. And I think that in particular, we're very committed in this account to recognizing the significant differences that obtain between levels of organization and the empirical fact that we have discovered such heterogeneity of developmental mechanisms that can underlie the same character, and trying to make sense of that in our account. So I think we're definitely, I think, more on the same page here than maybe at first glance. But Günter, did you want to follow?
Alan, you said it much more beautifully than I ever could. I rest my case.
I agree with that very much, but I think the frequent shifting between those levels of organization then somehow camouflages the explanandum from which you had set out.
But potentially, certainly those dangers are inherent whenever you move between levels in trying to work out theoretical ideas. So I take on board fully that that's a danger that we have to keep in view.
We have a question by Dana. Sorry, I'm not sure about the pronunciation.
Dana.
Yeah,
It's actually following up on what Gerd was saying. I was thinking as well about the methodological or strategic approach here, and thinking, yeah, it could raise worries that you're presenting a sort of reductive account where things are basically rooted in molecular evolution. So I liked hearing, yeah, that's not what's going on here, but it just makes me wonder how would you characterize the kind of strategy then? Are you starting with a recognition of these separate scales and then looking for sort of points where you can link them together? And I mean, one virtue of approaching things this way that I think James pointed out is when you do appeal to lower level mechanisms, that brings in a whole additional theoretical architecture, or which adds some new predictive power to the account. It opens up this kind of fruitfulness. But yeah, I guess I'm wondering, how carefully are you thinking about those kinds of moves you can make from one level to another, and is there a principled way of doing that?
Who wants to go first?
Günter, why don't you go ahead first?
And I'll leave things unsaid so that you can say something. Okay, so at one level, as a scientist, I'm of course committed to a sort of materialistic view of the world, right? So, in evolution is somehow related to molecular mechanisms, and that instantiates everything that we're talking about. And in the vast majority of cases, it's also genetic change that plays a role, regardless of whether first or second, or so, not going about, plasticity first and genes first. That's not but ultimately, molecular mechanisms are makes the machinery of life going, right? But that doesn't commit us to a reductive explanatory strategy where higher level phenomena like the identity of a cell type is in a reductive way explained by a particular form of molecular mechanism, right? And so what we try to do in all of our efforts in that area is to conceptually capture what's essential at a particular level of organization. Therefore, we're talking about character identity mechanisms, as Alan actually in his part said clearly, that are different kinds of mechanisms that contribute to individuality and different levels of organization, be them genes, cell types, tissues, organs, body parts, right? That's all, somehow related to some kind of, but each level of to molecular mechanisms, but each level of this of organization has its own kind of causal architecture that leads to the identity and recognizability of entities at that level of organization. So that is the way we try to thread the needle here, is to being materialistic in the basic outlook, but provide us a theoretical articulation that recognizes and sort of predicts in a way the fact that in different levels of biological organizations, unique ways of realizing character identity are in play. I don't know whether that makes sense, but that is.
Well, and I would just say that part of the way of taking seriously the complexity and the levels of organization is actually theorizing about them actively. And one of the advantages that we see in the character identity mechanism framework is that if you look at other accounts, so say ideas about deep homology, for example, there is a tendency to pay attention only to genetic regulatory networks and completely skip attention to tissues and organs and other levels of organization. So arguably, the vigilance that you need theoretically in these kinds of situations is to make sure that you're talking about these features explicitly. And we think that that's one of the things that we are attempting to do, at least.
Maybe an anecdotal historical context here. So the character identity mechanisms had a predecessor in character identity networks that I proposed a few years earlier. And so when Alan and I and other colleagues were together in Oslo, at one point, I think Alan pointed out that now people start to identify character identity with networks of gene regulatory networks, transcriptional, and then the conceptual alarm bells were ringing in all of our heads, and said, well, that it was just one example of how to think about character identity in a mechanistic way, but we shouldn't, it was my error to create the impression that everything has to be a gene regulatory network in the Davidsonian sense, and that was certainly not what we wanted. And we already played around with this idea that maybe for cell types it's not gene regulatory networks, but actual transcription factor complexes that we call quarks or core regulatory complexes. Is a different way of molecular way of thinking about cell type identity mechanistically, and which shows that it's gene regulatory networks in that sense is not applicable to all instances of parts of the body. So in a way, that the first paper that we did, the three of us together, was a way to repair that conceptual flaw in my own development, namely emphasizing too much one particular way of how development is regulated or controlled, and opening up the conceptual space to realizing that the evolution will find other ways of doing the same thing, and probably has to do it in different ways for characters at different levels of organization, right? So that the whole idea of character identity mechanisms was explicitly an anti-reductionist move. Mechanism is much more open than saying network, right? So I mean, that should hopefully lay to rest your worries in that respect.
Definitely giving me a clearer picture of what's going on.
Okay, I see there's a question. We're really running out of time. So Marie, do you have a very short question that could be followed by a very short answer, or this is not what you predict?
I can try to be as short as I can. So thank you for the talk first. I'm more a developmental biologist, so all the philosophy I'm trying to wrap my head around. One kind of, it's not really a question, it's more a problem and that I have, and I don't know how to fit it into what you said, is all the, for example, homeotic transformation, homeotic mutations. I don't know how often they happen actually in evolution of species and stuff, but I'm wondering, like in the very extreme case of Antennapedia, for example, in Drosophila, what are the legs that are in the antenna homologous to then? Because it's a completely developmental network then that's reused and co-opted for yeah. Sorry, I don't know if I'm clear, but maybe.
I think you're very clear, and the short answer is there is no short answer. It's actually one of the problems that we have kicked around the last few years, and I don't know, does James or Alan able to answer this quickly or more concisely?
I can maybe make a comment, but Garrett looks like he has a hand also. The ability to experimentally transform one character at one location into another is often taken as evidence of their serial homology, right? In some sense, right? So that's one bit We did write about this a little bit in one paper. I don't think it can be expressed very concisely, but maybe Gerd can shed light on this issue.
Go ahead, Gerd.
Gerd, is that?
Okay. I'm returning to something Günter has said, and I'm very kind of relieved about his expansion of the chins to the chimps, because it coincides very much with my notion of organizational homology, which is not just, as you mentioned, context dependency, but it implicates the progressive fixation of phenotypic architecture, which consists of discrete structural units, through increasing developmental interdependencies, not merely the genetic ones, but also the functional ones in the developmental dynamics that evolve.
Thank you for that comment. Yeah, that's a dimension that needs to be further developed, and I realize that our way of approaching it actually is probably a perspective that doesn't exclude additional ones, and some realization of that is already reflected in the fact that you can empirically individuate things out of context, but your notion is more complex because it also acknowledges that this reification of context can lead to another way of fixing character identities. Yeah.
Okay, so I think it's time to thank again our speakers. That was really fantastic and very inspirational. So thank you so much, Alan, Günter, and James, for this seminar. And I look forward to seeing you all in person, maybe in Bordeaux or elsewhere, whenever you want. You're invited.
Thanks for the opportunity to.
Yes, thank you, Thomas. Appreciated. Thanks everybody who was here.
Thank you so much. Bye-bye, everyone. See you soon.